Assalamu alaikum طلاب, and welcome to part 2 of our first lecture concerning the histology of the urinary system. Next part of our lecture will be discussing the histological structure of the nephron. As you know, the nephron is the structural and functional unit of the kidney. Each kidney in the human contain approximately 2 million nephrons, and these nephrons are the basic units that are responsible for the production of urine. Each nephron consists of two parts. In general, each nephron consists of two parts. The first part is this, the renal corpuscle, and it is the part of the nephron that's responsible for filtration of the plasma and formation of the initial ultrafiltrate, or called initial urine. Urine will then pass into a series of tubular structures within the cortex and within the medulla of the kidney, and these are called cortical and medullary tubular system. In today's lecture, we will discuss the renal corpuscle and how it's formed, what its structure, and in next lecture we will discuss the tubular system of the nephron. The renal corpuscle, as you can see here, is the initial dilated part of the nephron. This is called the renal corpuscle. It's about 200 micrometer in diameter, and it's the site for the initial filtration of a plasma to produce the glomerular ultrafiltrate. This structure, that's to say the renal corpuscle, is always located within the cortex. Within the renal corpuscle, we can see the glomerulus, which is a tuft of capillaries formed of about 10 to 20 capillary loops surrounded by a double-layered epithelial capsule called the renal capsule or Bowman's capsule. This is the tuft of capillary, and as we discussed in part 1 of our lecture, this tuft of capillary is formed by branching from the efferent arteriole within the renal corpuscle. So here we have the efferent arteriole, which will branch into several capillaries, and these capillaries will merge together then to form the efferent arteriole. The best way to understand how the glomerulus is formed and how the renal corpuscle have developed is to imagine the tubular system as a closed-ended tube where its end is spherical and distended. The tuft of capillaries of the branching from the efferent arteriole will be introduced into this spherically distended sphere. This will cause some of the cells closely coating the capillaries, and these cells are called podocytes, or the visceral layer of the capsule. However, this layer is also continuous with the cells lining the Bowman's capsule, or the parietal layer of the capsule. So, Bowman's capsule is this spherical structure, and it is composed of two layers. We have the internal layer or visceral layer that closely envelop the glomerular capillaries, and we have the parietal layer that form the surface of the capsule. Between these two layers, we have this space here, and it's called the capsular space or urinary space. As it is evident from this photo, each renal corpuscle has two poles. The first is the vascular pole, where we have the afferent arteriole and efferent arteriole entering and exiting from the corpuscle, and we have the other pole is the tubular pole, which is continuous with the proximal convoluted tubule. Here we have a section of the kidney to further illustrate what we have said earlier about the nephron and the histology of the kidney. This section is taken from histologyguide.com, the website that we use to study the practical histology. As you can see here, it's very evident that the kidney is divided into inner medulla, this is the medulla, and outer cortex. So we have inner medulla and outer cortex. The cortex, within the cortex, we have these spherical structures. These are the renal corpuscles. These spherical structures are the renal corpuscles. And we have different types of nephrons depending on the situation of this renal corpuscle within the cortex. We have cortical, subcapsular nephrons, where the renal corpuscle are located below the capsule. We have mid-cortical nephrons here in this area, and we have juxtamedullary nephrons here in this area. We can also see between the renal corpuscles, we can see this areas, this striation, which is called the medullary rays. Okay, this is a higher magnification of one corpuscle, and it's very evident here. This is the tuft of capillary that we call it a glomerulus. The Bowman's capsule surrounding the glomerulus, and it's composed of two layers. The layer immediately surrounding the capillaries are called the visceral layer, and the layer that form the outer part of the Bowman capsule is called the parietal layer, and between them we have this space which is called the urinary space. Vascular pole is here, where the afferent and efferent arterioles are entering and exiting the glomerulus. The renal corpuscle is very important because it contains the filtration apparatus of the kidney, where plasma is filtrated initially. This apparatus is composed of a complex semi-permeable barrier called the glomerular filtration barrier. This barrier is highly permeable for water and small and medium-sized molecules. However, it is impermeable to serum albumins and other larger proteins. So, it is abnormal to see serum albumin or larger proteins in the ultrafiltrate or in the urine. Also, it is abnormal to see red blood cells and white blood cells within the urine. However, the filtrate usually contain water, glucose, amino acids, urea, many hormones, vitamins, and other small molecules. These can be seen normally within the filtrate. The glomerular filtration barrier is composed of three important parts. First is the fenestrated endothelium of the glomerular capillary. Second is the glomerular basement membrane, and third is the visceral layer of Bowman's capsule. As it is evident from this photo, here we can see the glomerular capillary, which is highly fenestrated. The fenestrations within this capillary is more numerous and wider, larger, and more irregular than any other fenestrated capillary in the body. It also contains no diaphragm for the ease and rapid passage and transport of water and other molecules. The endothelial cells here contain transmembrane protein called aquaporin, which is a water channel to allow the rapid filtration of water into the Bowman's capsule. The second component of the glomerular filtration barrier is the glomerular basement membrane. This is a thick basement membrane, as we can see here. It's formed by both the endothelial cells and the podocytes. Its thickness is about 300 to 370 nanometer in thickness, and it's formed by different types of collagen and proteoglycans and glycoprotein. These molecules will act as a meshwork to restrict the passage of large molecules or large protein from the plasma. So any protein larger than 70 kilodalton cannot be passed through this barrier, and it will be retained within the plasma. However, smaller proteins and smaller molecules can be passed through this membrane, and the protein especially will be degraded and then removed by the proximal convoluted tubule. The third part forming the glomerular filtration barrier is the visceral layer of Bowman's capsule. And as we said earlier, the visceral layer of Bowman's capsule envelop and surround the glomerular capillaries. It is formed of a specialized type of cell called podocyte. These podocytes composed of cell body, and it will send several processes to surround the glomerular capillaries. As we can see here, the podocyte, which is the visceral epithelial cell, is composed of cell body sending several primary process, and each process will send a secondary process that will interdigitate with the secondary processes of other podocyte cell. Here, a scanning electron microscope showing a podocyte with the primary processes and secondary processes. These secondary processes, between the secondary process, we have a space that's called filtration slit, through which the filtration of all the materials will occur from the plasma toward the urinary space. So this is an electron microscope showing the podocyte cells. This is a podocyte, this is a podocyte, and here we have the glomerular capillary, and we can see how the foot processes of the podocytes are surrounding and enveloping the glomerular capillaries. These are the foot processes, and between these foot processes, there are spaces that are about 40 nanometer in width, called filtration slits, and through these spaces the filtration of substances will occur. These filtration slits are covered by specialized intercellular junctions known as slit diaphragm. Here it is very evident the filtration apparatus of the kidney. Here we have the glomerular capillary. This is the endothelial cells, which has the fenestration inside it between the endothelial cells, and we have the basement membrane, glomerular basement membrane, and we can see that it's a very thick basement membrane, and we have here the podocytes and its foot processes and filtration slits and slit diaphragm. This arrangement of the glomerular filtration apparatus is very important, because any disruption or any dysfunction of this apparatus will cause kidney or renal disease. Podocytes are very important, and abnormality in podocytes, and especially in the foot processes of the podocytes, can be seen in many renal diseases. And the first manifestation of abnormality in the foot process of the podocyte will be the effacement or retraction of the foot processes. That's to say, the foot processes of the podocytes will not completely surround the glomerular capillary. This will lead into disruption of the filtration apparatus and abnormal infiltration, and we can see in many kidney diseases that we have a loss of large size proteins, we have loss of RBCs and white BCs into the urine, and these are all important features that can be seen especially in cases of renal failure. So to recap, the filtration apparatus of the kidney is composed of three important components. First, we have the endothelial cells surrounding the glomerular capillaries. These glomerular capillaries are characterized by being highly fenestrated without diaphragm. The fenestrations are large and irregular. The second component is the glomerular basement membrane, which is a thick basement membrane, as we can see here. And the third component of the filtration apparatus is the podocytes and its primary, secondary, and the foot processes of the podocytes. filtration will occur from the plasma toward the urinary space in the Bowman's capsule. And of course, this is the parietal layer of Bowman's capsule. In the final part of our lecture, we will discuss the mesangium. Mesangium is an important component of the renal corpuscle, and it's formed from mesangial cells and their extracellular matrix. Mesangial cells and the mesangial extracellular matrix is very obvious at the spaces between the adjoining glomerular capillaries. So these spaces between the glomerular capillaries are filled with a special type of cells and special type of extracellular matrix called the mesangium. The mesangial cells and its matrix also can be found within the vascular pole of the renal corpuscle. That's to say, it's outside the renal corpuscle, surrounding the afferent and efferent arterioles, and where they form part of what is called the juxtaglomerular apparatus that we will discuss in the next lecture. These mesangial cells and its mesangial matrix are enclosed by the same basal lamina of the glomerular capillaries. The main function of mesangial cells and mesangium is to give physical support to capillaries within the glomerulus. They also help to maintain the optimal filtration rate, and they have a phagocytic function, especially of protein aggregates and antigen antibody complexes that can be seen in many pathological conditions within the mesangium, and they secrete several cytokines and prostaglandins and other factors important for immune defense and the repair of the glomerula. So to summarize our lecture, we will summarize it through histological sections and pictures. Here we have the kidney, and it is divided into cortex, outer cortex, and renal inner medulla. The medulla is formed as a triangular structure called renal pyramids, and we have about 8 to 12 renal pyramids in each kidney. Between the renal pyramids, we have sections of cortical tissue extending deep into the kidney, and these are called renal columns. And within the cortex, we have these striations that are called medullary rays. These medullary rays extend from the medulla into the cortex. Within the histological section, we also see the cortex and the medulla, and this is the corticomedullary junction. The dashed line is the corticomedullary junction, or here we can see it in this area. The base of the medullary pyramid is the corticomedullary junction. The cortex, the most important histological structure within the cortex is this spherical structures that are called renal corpuscles. Renal corpuscles, and we have these collection of tubes and tubules extending from the medulla into the cortex. These are called medullary rays. A higher magnification of the renal cortex, and we can see here the renal corpuscles, and as we said earlier when we discussed the renal corpuscles, are only found within the cortex. We cannot find a renal corpuscle within the medulla. The renal corpuscles are only seen in the cortex of the kidney. So these are the renal cortex, the renal corpuscles, and these are the medullary rays. Each renal corpuscle is formed from a tuft of capillaries called the glomerulus. This is the glomerulus surrounded by Bowman's capsule. Bowman's capsule is formed of two layers. We have the visceral layer that immediately surround the capillary, and we have the parietal layer. The visceral layer is also called the podocytes. These are specialized cells surrounding the renal the glomerular capillaries and are called podocytes. We have the space which collect the ultrafiltrate. This is called the urinary space. And each renal corpuscle has two poles. We have vascular pole through which the afferent and efferent arterioles enter into the glomerulus, and we have the tubular pole that connect with the proximal convoluted tubule. And by this we reach the end of our first lecture of the histology of the urinary system. Thanks for your listening.